Moving on to the more functional aspect of neurons. So in order to send an electrical signal down the plasma membrane of your axon, there has to be a flow of electricity. So first point for a resting cell, you have an electrical potential. So it's a difference in the concentration of charged particles between one point and another. So living cells have an electrical potential, so a difference in charge between the inside and outside of the cell, and that's called the resting membrane potential. The cells tend to have more negative particles on the inside, so some of your proteins, as well as with your sodium-potassium pump, adjusting the levels of your ions inside and outside your cell. So for neurons, their resting membrane potential is about negative 70 millivolts. So if you remember from the muscle chapter, it was negative 90 for your muscle cells. So for neurons, it's a little bit different. It's a resting membrane potential of negative 70. So when you move charged particles in and out the cell, it causes an electrical current. So this flow will change your membrane potential and therefore causing other effects to happen inside your cell. So starting at rest, we have a membrane potential of negative 70 millivolts. So again, the outside of the cell is more positive, the inside of the cell is more negative. You have more anions that can escape, particularly, for example, proteins are negatively charged, helping to create the more negative charge on the inside of your cell. And again, with your sodium-potassium pump, also affecting only about 3 millivolts of that charge, but sending excess sodium ions out the cell and bringing in an excess of potassium ions inside the cell. Here is a chart looking at the difference in change or difference in charge during an action potential. So same thing like your muscle cells, a quick depolarization followed by a quick repolarization causes is what's considered your action potential. There's an extra part for your neuronal action potential, and that's a small section here called a hyperpolarization. So still having local potential where there's a slight change in the cell, eventually reaching threshold. During depolarization, you have a rush of sodium into the cell, repolarization, rush of potassium out the cell, so going towards something positive, and then now we're going back towards our negative. And then you eventually overshoot your resting membrane potential, which is here. That overshoot is called hyperpolarization, and what that does is it prevents your neuron from sending another signal too quickly. So unlike your muscle cells, your neuronal cells cannot end up with a temporal wave summation or a fused tetanus due to too quick of stimulation. The hyperpolarization prevents your neuronal cell from sending another active potential. So it essentially slows it down to make sure that your cell goes all the way back to resting membrane potential. Okay, so the first step in our action potential again is depolarization. So once you reach enough stimulation for the difference in charge to reach your threshold part, so that's when your voltage-gated sodium, which are fast, sodium channels which will open quite fast, will open up, and your voltage-gated potassium channels will also open up, but they're so slow that by the time they actually do open up, that's at the peak of your action potential. So first step again, depolarization, sodium rushing into the cell, going down their concentration gradient, as well as being attracted to the negative charge on the inside of the cell. Eventually, your sodium channels will start to close, and at this time, your voltage peak is about 35 millivolts. By this time, your slowly opening voltage-gated potassium channels will finally decide to open. And when they do that, the potassium rushes out the cell, going down its concentration gradient, as well as being repelled by all this positive charge inside the cell, and therefore causing the inside of the cell to become more negative. Eventually it gets to the point where your cell becomes hyperpolarized, so more negative than our resting membrane potential, shown here on this end. and that causes the refractory period, so it prevents your sodium channels from opening up again, so preventing this whole action potential process. And then eventually your resting membrane potential is restored. So there's some characteristics of an action potential. It follows a all or none law. So it's going to shoot an action potential or you're not going to shoot an action potential. So as long as you reach threshold, you will in fact always will fire off an action potential. If you don't reach threshold, it's not going to happen. Basically, that's it. And once it starts, it can't stop. So it's not like you can start sending an action potential down your axon and then all of a sudden your neuron changes its mind and stops the action potential from going. It's going to go all the way down to the end part of the neuron. Once you reach the end of your neuron, then that electrical signal that was traveling down the axon is then converted into a chemical signal into what's called a neurotransmitter. So a neurotransmitter is now our chemical signal, be able to send that message to our next cell. So at the synapse, that action potential will trigger the release of the neurotransmitter from vesicles that are located in that synapse. And then once this neurotransmitter is released across that gap in the synapse to the next cell, then it stimulates a new wave of electrical activity into the next cell. So it's kind of like passing off the baton to another runner. It's just playing tag. Okay, you're up next. Continue to send this signal. So there's two neurons involved in a synapse. You have your presynaptic neuron is the one that's going to release the neurotransmitter. Then your second neuron is your postsynaptic neuron that will respond to that neurotransmitter. So the presynaptic neuron will synapse onto the target cell. So if you're just focusing on another neuron, either through different parts of the neuron, so it can synapse on the dendrite, it can synapse on the neurosoma, it can synapse on your axon, or on from the axon to your another axon. So there's different locations for getting the signal. Mostly it's going to be in your dendrite region as well as in your neurosoma region. So for example, you can have a ton of synapses between cells. So for example, in a spinal motor neuron, so sending a signal from the spinal cord to a target motor cell, can have about 10,000 axon terminals, most of them being in their dendrites, about 8,000 there, and about 2,000 ending in your neurosoma. In the cerebellum, when we get to the brain chapter, the way the neurons are organized in your cerebellum, so that's the little wrinkly part at the back of your brain, the way those cells are organized, there's a lot of connections. So one neuron can have as many as 100,000 synapses just because of how it's so tightly packed and organized in the cerebellum. For your synapse, the distance between your presynaptic neuron and your postsynaptic neuron is called your synaptic cleft. So this is the small distance that your neurotransmitter chemical signal will travel between the two cells. This distance was found out by Ramon E. Cajal. So originally we thought neurons touched each other, and then we found out later that they actually don't. He figured this out through a experiment looking at the use of chemical messengers through an experiment that's described here, but you don't have to know that. The chemical messenger that he was focused on, since he was dealing with the heart being a muscle cell, the neurotransmitter for muscle cell is acetylcholine. He named it vagus substance. Later it was renamed to acetylcholine. So it's our first known neurotransmitter, and for some reason it's only by itself, it's the only neurotransmitter that is structured the way it is. So it's in its own special little category. So here's a cute little cartoon version of a chemical synapse. So at the top we have our presynaptic neuron. Here's our axon traveling down towards our synapse. So there's our electrical signal. It's going to send the signal down, and then once you reach the axon terminal, what's not shown here, but I'm going to say it anyway, that action potential will trigger voltage-gated calcium channels to open, allowing calcium into the cell, which causes these synaptic vesicles, so our little blue bubbles looking here, to merge with the plasma membrane, releasing the neurotransmitter into the synaptic cleft. And then the neurotransmitter goes from high concentration to low concentration, eventually being pushed out into your space and binding to the receptor. So the neurotransmitters are ligand, and it's going to bind to a receptor on your postsynaptic neuron. So there's multiple types of neurotransmitters. So we already covered acetylcholine being by itself, usually in your muscle contractions. You also have neurotransmitters that are made of amino acids, including one that we'll talk about again later called GABA or gamma. So there's your G, Greek letter G. Aminobutyric acid, no one really says all that. We just say GABA. That's actually an inhibitory neurotransmitter. You have other neurotransmitters, so your monoamines, so it's made from amino acids. So these include your epinephrine, norepinephrine, also called your adrenaline and noradrenaline, released from the adrenal glands that are above your kidneys, so affecting the action in your body that way. Your dopamine, which is released during pleasurable activity. Also your histamine, as well as ATP and serotonin can be a type of neurotransmitter. Then lastly, you have your gases and neuropeptides, again, being able to trigger a response in the next cell. So with your postsynaptic neuron receiving a signal from your presynaptic neuron, it's not always stimulatory. It either can be excitatory to help to increase that local potential and going towards a threshold, or it could be inhibitory, making the inside of the cell more negative and therefore away from your threshold. So again, this depends on the type of receptor. So your neurotransmitter is your ligand. It will bind to the receptor, which works as a gated ion channel, or it can also work as through second messengers. So there's three different types of synapse that we'll talk about. We have two excitatory and one inhibitory. We have the excitatory cholinergic synapse, the inhibitory GABAergic synapse, and your excitatory adrenergic synapse. So an excitatory cholinergic synapse, your acetylcholine is released from your presynaptic neuron, binds to a receptor in your postsynaptic cell. So this allows positive ions to move into that neuron or next cell. And as you have these positive ions moving that local potential towards threshold, and more likely to fire an action potential. Okay, your next two synapses don't have pictures. Oh well. For your GABAergic synapse, so your GABA neurotransmitter will bind to a chloride channel, chloride being a negative ion. So when that gated channel opens due to the ligand gamma binding to it, negative chloride ions flow into the cell. When those negative chloride ions move into the cell, it makes the inside of the cell more negative, and therefore moving the membrane potential of that cell away from threshold, and therefore less likely to fire an action potential. Your last type of synapse is your excitatory adrenergic synapse. So your norepinephrine, also called your noradrenaline, works through second messengers. So remember our cyclic AMP from way earlier in the semester. Basically will have a little bit slower response, so not immediate like you have with those ion channels, but it helps to helps to amplify the effect of the simple ligands. And this one actually does have a picture, I forgot, sorry. Okay, so binding to our G protein coupled receptor, causing CAMP to be made, and then making a whole bunch of other effects in the body. Eventually, what's made is a ligand that will open a ligand-gated ion channel, in this case sodium, so positive ion going into the cell, causing the local potential to increase and going towards threshold, and therefore more likely to fire. So we've covered how to start a signal inside of the postsynaptic cell. Now we have to stop it. So in order to turn off the stimulation, there's multiple ways to do so. So first one, your presynaptic cell can stop releasing neurotransmitter. So no new neurotransmitter being able to bind to that ligand, neurotransmitter binding as a ligand to the receptor helps to stop the signal from continuing in the postsynaptic neuron. So for example, acetylcholine, so your neurotransmitter only stays for about 1 millisecond and then it leaves that receptor. So for example, your acetylcholine gets broken down by acetylcholine esterase. So once it removes itself off the receptor, then to prevent the acetylcholine from coming back and binding to another receptor, acetylcholine esterase breaks it down, and then those bits and pieces and parts are taken back into the presynaptic cell where they're repackaged as acetylcholine and then resent back down through fast axonal transport, so with anterior grade transport, sending the materials back down to those axon terminals so they can be re-released again. Another way to do this is to reabsorb the neurotransmitter. So just having channels that are available in the presynaptic cell, taking in your neurotransmitter to prevent it from binding yet again to a receptor. Next way to stop the signal is to diffuse into the extracellular fluid. So as your neurotransmitter is moving from high to low concentration, eventually it's just going to diffuse out of that space. So no longer able to bind to the ligand. So here's a little picture version of it. So like for example, acetylcholine being released, but then it's also broken down to acetylcholine esterase. So stop that signal. Sometimes you can just diffuse away, and therefore you're not able to continue to have the signal to continue to bind to that receptor. So that's it for Chapter 12. For the rest of the semester, we'll actually be focusing on the nervous system. So you're kind of in for the ride for the rest of the semester. But it's still